Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the intellectual landscape of early 19th century France, the human mind was a territory mapped by conjecture and anecdote. The dominant cartography was phrenology, a compelling and widely popular system conceived by Franz Joseph Gall. This doctrine proposed a direct and granular correspondence between the contours of the human skull and the landscape of human character. Specific faculties, from benevolence and conscientiousness to combativeness and amativeness, were believed to reside in distinct cerebral organs, their development supposedly mirrored by the palpable prominences of the bone encasing them. Phrenology offered a seductive sense of order, a tangible key to the complexities of personality and intellect. Its profound weakness, however, was its foundation upon selective observation and its complete insulation from empirical refutation. The system was a closed loop of confirmation, a science of touch and tail that lacked a crucial element, a method for experimental verification. Into this environment stepped Pierre Flourens, a physiologist armed with a profound skepticism and a surgeon's scalpel. He recognized that the grand claims of phrenology, and indeed any theory of brain function, would remain in the realm of philosophy until a rigorous, repeatable, and causal methodology could be brought to bear on the living brain itself. The challenge was immense. The brain was the organ of sentience and volition, and investigating it required a technique that could isolate its components to observe their unique contributions to the whole. Flourens's answer to this challenge was the systematic development and application of experimental ablation, a procedure also known as localized lesioning. This was his great methodological innovation. The approach was direct and conceptually powerful. By precisely removing a specific part of the brain in an animal model, and then carefully observing the resulting deficits in behavior, one could deduce the function of the excised part. His seminal one is a fourth publication, titled Recherches expérimentales sur les propriétés et les fonctions du système nerveux dans les animaux vertébrés, or Experimental Researches on the Properties and Functions of the Nervous System in Vertebrate Animals, laid out as neuroscience. The work was a masterpiece of methodical clarity, detailing experiments primarily on pigeons and rabbits, animals whose behavioral repertoires were complex enough to show deficits yet manageable enough for laboratory observation. The choice of animal was deliberate. The goal was to identify fundamental principles of nervous system organization common to vertebrates. A quintessential example of this technique, one that struck a decisive blow against the speculative nature of phrenology, was his investigation of the cerebral hemispheres. Consider the elegant and revealing simplicity of the procedure. After the careful surgical removal of the cerebral hemispheres from a pigeon, the animal underwent a profound transformation. It was alive, yet it appeared to have lost its connection to the world. The bird could stand and, if pushed, could walk. It could be tossed into the air and would flap its wings. Its basic motor machinery and spinal reflexes were clearly intact. What had vanished was the internal impetus for action. The pigeon displayed a complete absence of volition, memory, and perception. It would starve to death in front of a pile of grain because the recognition of food and the will to eat were gone. It would fail to react to a threat until those stimulus became a direct physical assault. The creature was reduced to a biological automaton, a living collection of reflexes without a mind to guide them. The conclusion was inescapable and profound. The higher faculties of will, judgment, and perception were functions of the cerebral hemispheres. Flourens had established a causal link between a specific anatomical structure and a complex set of psychological functions. This was a world away from Gall's practice of associating a penchant for poetry with a bump above the eye. Flourens had created a reproducible experiment that yielded clear, observable results, establishing a standard of evidence that would define the future of neuroscience. He demonstrated that one could ask a direct question of the nervous system and receive a comprehensible physiological answer. Flourens's work was the culmination of a powerful current in French science. He operated within a vibrant tradition of experimental physiology, a lineage extending from the meticulous spinal cord investigations of Julien-Jean César Legalwar. Legalwar had earlier demonstrated, through lesioning experiments, that the fundamental center for respiration was located in the medulla oblongata, proving that a vital and complex function could be pinpointed to a small region of the nervous system. Flourens's approach was also philosophically fortified by the unapologetic empiricism of his contemporary, François Magendie, a towering figure who championed vivisection as the essential, primary tool for physiological discovery. Magendie's own famous experiments, which clarified the distinct sensory and motor functions of the dorsal and ventral spinal nerve roots, were a testament to the power of direct intervention in revealing biological truths. Flourens took the principles advanced by these predecessors and applied them with unprecedented focus and systematic rigor to the most complex organ of all, the brain. The legacy of this methodological breakthrough was immediate and enduring. Flourens forged the essential tool for a new science. The logic of his method, the careful correlation of an observed functional deficit with a specific, localized anatomical lesion, became the conceptual blueprint for generations of neurologists and physiologists. Its influence is perhaps most clearly seen in the work of Paul Broca decades later, when Broca encountered his patient Louis Victor Labortny, a man who could understand language but whose speech was reduced to the single-syllable tan. He was observing a human experiment of nature. Upon Labortny's death, Broca's autopsy revealed a distinct lesion in a specific area of the left frontal lobe. In linking this anatomical damage to the precise language deficit, Broca was applying the very same lesion deficit paradigm that Flourens had pioneered in his pigeons. He was translating Flourens's experimental logic from the animal laboratory to the human clinic, thereby confirming the principle of functional localization within the human cerebrum with profound consequences for our understanding of language and the mind. With the invention of experimental ablation, Pierre Flourens had provided more than a critique of phrenology. He had supplied the foundational methodology upon which the entire edifice of modern brain science would be constructed. He had turned the brain from a subject of speculation into an object of systematic investigation. While Paul Broca would later apply this powerful lesion deficit logic to the human cerebrum, the principle's most decisive and elegant demonstration came from Flourens himself in his exploration of a different, deeply enigmatic region of the brain. For centuries, the cerebellum, the smaller structure nestled beneath the great cerebral hemispheres, remained a profound mystery. Anatomists had described its intricate, tree-like internal structure, the arbor vitae, but its purpose was the subject of wild and unsubstantiated conjecture. Theories attributed to it everything from the seat of memory to the source of animal passions. The Italian anatomist Luigi Rolando had performed some early, cruder experiments involving cerebellar injury, but his results were ambiguous and his conclusions lacked the clarity needed to settle the debate. The intellectual landscape was a fog of speculation, awaiting the sharp, clarifying light of a definitive experiment. It was into this fog that Flourens directed his scalpel with unparalleled precision. His chosen subjects, once again, were pigeons and other animals whose motor behaviors were easily observable and quantifiable. The genius of his method lay in its surgical tidiness and the focused nature of his intervention. He developed techniques for exposing the cerebellum with minimal damage to surrounding tissues, allowing him to perform a clean, targeted ablation. He would meticulously remove the cerebellum, layer by layer or in its entirety, and then closely observe the animal's subsequent behavior. The results were immediate, dramatic, and utterly unambiguous. An animal deprived of its cerebellum underwent a startling transformation. Its capacity for sensation appeared intact. It would react to a touch or a loud sound. Its intelligence and volition, the faculties Flourens attributed to the cerebrum, also seemed preserved. The pigeon would still attempt to escape or seek food. Its muscles retained their full contractile power. Yet, the creature was rendered totally helpless. Every attempt at purposeful movement dissolved into a storm of chaotic, ineffective action. If it tried to walk, it stumbled, staggered, and fell, its legs moving with a clumsy, exaggerated gait. If it tried to fly, its wings beat the air in a frantic, asynchronous flapping that produced violent tumbling instead of lift. The animal became a living portrait of incoordination, a body whose constituent parts had lost their conductor. Flourens described these effects with the dispassionate accuracy of the master observer. He saw that the will to move persisted, and the muscular machinery to execute the movement was fully functional. The link between intention and coherent action was severed. The animal became a vessel of disorganized impulses, unable to regulate the force, timing, and direction of its own limbs. It was a marionette whose strings were being pulled randomly, without a guiding hand. Through these meticulous excisions, Flourens provided the first irrefutable evidence that the cerebellum orchestrates the intricate symphony of voluntary movement. It was a central governor of equilibrium and the harmonizer of motor activity. This discovery was a monumental piece of physiological insight, replacing speculation with a concrete, experimentally verified principle of neural function. He had isolated a specific, complex function, motor coordination, and pinned it directly to the specific anatomical structure, the cerebellum. The clarity of this finding provided an unshakable cornerstone for the burgeoning field of neurology. It gave clinicians a framework for understanding a host of human movement disorders. The staggering gait, slurred speech, and trembling movements seen in patients with cerebellar damage, a condition now known as cerebellar ataxia, could be understood as a direct consequence of the loss of the very function Flourens had identified in his pigeons. His laboratory experiments provided the fundamental explanation for clinical reality, bridging the gap between basic animal physiology and human medicine. The precision of his work gave it an authority that was difficult to challenge, and the role of the cerebellum in motor control became one of the first widely accepted tenets of modern brain science. Furthermore, The legacy of this discovery extended far beyond its immediate clinical utility. It shaped the very course of subsequent neurological research. Flourens had functionally demarcated a piece of neural territory. Later investigators, armed with new tools, could now explore this territory with a map in hand. A prime example is the work of the Scottish neurologist David Ferrier a few decades later. Ferrier pioneered the use of localized electrical stimulation as a method for exploring brain function, while Flourens used ablation, the removal of tissue, to see what function was lost. Ferrier used stimulation to see what action was produced. When Ferrier applied his electrodes to the cerebellum of monkeys, he found he could elicit complex, coordinated movements of the eyes, head, and limbs. He was essentially activating the very coordinating system that Flourens had deactivated. Ferrier's work was a direct extension of Flourens's, building upon the foundational knowledge of the cerebellum's role. Flourens had identified the what, the cerebellum's function, providing the essential context for Ferrier to investigate the how, the specific pathways of its influence. In his investigation of the cerebellum, Pierre Flourens achieved a perfect synthesis of experimental rigor and conceptual clarity. He took a structure shrouded in anatomical mystique and rendered its primary function transparent. This particular achievement stands as a testament to the power of his experimental method when applied to a system with a relatively localized and distinct role. He had provided an unassailable piece of the intricate puzzle of brain function, a foundational truth that would anchor future exploration and withstand the test of time. This triumph of localization, however, would stand in stark and fascinating contrast to the grand, overarching theory he would champion for the brain's most complex and commanding structure, the cerebral hemispheres themselves. Flourens's meticulous investigation of the cerebellum provided a firm foundation for understanding a specific, localized brain function. This success, however, served as a compelling counterpoint to his subsequent and ultimately less successful theory concerning the cerebral hemispheres. His work on the cerebellum showcased the power of precise experimentation in revealing the distinct roles of specific brain regions. This precision, however, contrasted sharply with the holistic model he proposed for the cerebrum, a model that, while initially influential, would ultimately prove to be an impediment to further progress in neuroscience. The problem Flourens confronted was monumental: to construct a model of cerebral function that could reconcile his experimental findings with the prevailing, yet flawed, understanding of the brain. Phrenology, with its simplistic assignment of discrete mental faculties to specific cortical regions, had fallen out of favor due to its lack of empirical support. Flourens, through his ablation experiments on pigeons, aimed to provide a more accurate, albeit dramatically different, understanding. His method involved carefully removing portions of the cerebrum and observing the resulting behavioral changes. He noted a consistent pattern of lesions, unless extensive, produced a generalized impairment of higher cognitive functions: will, judgment, perception, rather than a selective deficit in a single faculty. Furthermore, he observed a remarkable capacity for functional recovery over time, provided the damage was not too widespread. These observations led him to formulate his doctrine of asterisk action, commune asterisk, a concept synonymous with equipotentiality. The core tenet of this doctrine was that the cerebral hemispheres function as a unified, indivisible entity. Flourens famously asserted that the capacity for sensation, perception, and volition constituted a single, fundamental faculty. This assertion was far from a purely empirical conclusion. It was deeply intertwined with his philosophical leanings, specifically his adherence to a Cartesian dualism that posited an indivisible soul. This philosophical perspective heavily influenced his interpretation of the experimental data, leading him to envision the cerebrum as a unified organ reflecting the unity of the mind. René Descartes' influence on Flourens's interpretation is undeniable. Descartes' concept of the soul as an indivisible entity provided a ready-made framework for understanding the seemingly holistic nature of cerebral function as revealed by Flourens's experiments. This philosophical predisposition likely played a significant role in shaping his interpretation of the experimental results, leading him to emphasize the unity of function over the possibility of localization. The immediate impact of Flourens's doctrine was profound, yet ultimately restrictive. His considerable authority and the seemingly compelling nature of his experimental findings established a powerful anti-localizationist dogma that significantly hampered the progress of neuroscience for several decades. This dogma held sway over the scientific community, creating a considerable obstacle for researchers who sought to demonstrate the localization of specific functions within the cerebral cortex. The case of Paul Broca provides a particularly striking illustration of this intellectual barrier. Broca's discovery of a specific area in the left frontal lobe crucial for speech production. Broca's area directly contradicted Flourens's holistic view. The acceptance of Broca's findings required a substantial amount of evidence and a significant shift in the prevailing scientific paradigm, a shift that was delayed considerably by the enduring influence of Flourens's doctrine. Even into the 20th century, the legacy of Flourens's holism persisted. Leslie's concept of mass action, which posits that the extent of behavioral impairment following cortical lesions is proportional to the amount of tissue destroyed rather than the specific location of the damage, can be seen as a direct intellectual descendant of Flourens's equipotentiality. While Leslie's work refined and modified Flourens's original theory, it nevertheless maintained the fundamental principle of the cerebrum's integrated, holistic functioning. However, the critiques of Flourens's doctrine were not absent. Many researchers questioned the generalizability of his findings from pigeons to humans, noting the considerable differences in the structure and complexity of the avian and mammalian brains. Furthermore, the functional recovery Flourens observed might have been due to compensatory mechanisms within the brain rather than an inherent equipotentiality. These critiques, along with the accumulating evidence for cortical localization, gradually eroded the dominance of Flourens's holistic view. Despite its eventual refutation, Flourens's theory of cerebral holism played a crucial, albeit ultimately hindering, role in the history of neuroscience. His work, while flawed in its central conclusion, stimulated considerable research and debate, ultimately contributing to a more nuanced and accurate understanding of brain function. The legacy of his doctrine serves as a poignant reminder of the interplay between experimental observation, philosophical interpretation, and the complex evolution of scientific thought. His influence, both positive and negative, remains a compelling case study in the history of scientific progress. The story of Flourens's holistic doctrine stands as a testament to the enduring challenge of interpreting experimental data within a complex and ever-evolving scientific landscape. Flourens's contributions extended far beyond his controversial theory of cerebral equipotentiality. His experimental prowess yielded significant advancements in our understanding of several crucial physiological processes, often overshadowed by the intense debates surrounding his cerebral localization views. A prime example is his meticulous work on the identification of the vital knot, the noid vital asterisk, a pivotal discovery that refined our understanding of respiratory control, building upon the groundwork laid by Legalvar, who had suggested a crucial respiratory center within the medulla oblongata. Flourens employed a remarkably precise experimental approach. He meticulously created highly localized lesions in the medulla of experimental animals, systematically mapping the effects of these lesions on respiratory function. This was no mere crude ablation. Flourens's precision was remarkable for his time. He did not simply destroy large sections of tissue. Instead, he targeted incredibly small areas, employing instruments of extraordinary refinement to achieve pinpoint accuracy. The consequence of this meticulous approach was the identification of a specific, minuscule point within the medulla whose destruction resulted in an immediate and irreversible cessation of breathing. This pinpoint location, previously unknown, became known as the noid vital asterisk, the vital knot, the primary respiratory center. This discovery provided an unparalleled level of anatomical and functional precision to the understanding of respiratory control, a significant advancement that directly influenced subsequent research on the brainstem and its vital functions. Physiologists following Flourens built upon his findings, refining the understanding of the neural circuitry involved in respiration, eventually leading to a more complete picture of the intricate mechanisms maintaining this critical life function. His work served as a foundation for later investigations into the specific neuronal populations and neurotransmitters involved in respiratory rhythm generation. Flourens's influence extended into the realm of anesthesiology, a field then in its infancy. The introduction of ether and chloroform as surgical anesthetics presented a significant medical advance, yet their physiological mechanisms remained largely enigmatic. Flourens's experimental work provided a crucial framework for understanding their effects. In 1847, he published observations detailing the sequential action of these agents on the central nervous system. His experiments showed that these anesthetic compounds initially affected the cerebral hemispheres, resulting in the loss of consciousness. Increasing the dosage then affected the cerebellum, leading to a loss of coordination and motor control. Finally, at sufficiently high concentrations, these agents fatally depressed the noid vital asterisk, causing respiratory arrest. This sequential action, meticulously documented by Flourens, provided a critical physiological basis for the safe clinical administration of anesthetics, a direct and immediate impact on surgical practice. It provided surgeons with a deeper understanding of the potential risks and a rational basis for titrating the dosage to achieve effective anesthesia while minimizing the risk of respiratory depression. This work significantly contributed to the development of safer anesthetic techniques and greatly improved surgical outcomes. The detailed understanding of the sequential effect of anesthetic agents on different brain regions, directly derived from Flourens's experiments, became the cornerstone of anesthesiology, education, and practice, influencing generations of physicians. Another remarkable contribution lies in Flourens's work on the semicircular canals. By creating lesions in these inner ear structures of pigeons, he observed pronounced deficits in balance and equilibrium, a discovery that revolutionized our understanding of the vestibular system. These experiments clearly differentiated the function of the semicircular canals from hearing, establishing them as the primary peripheral sensory organ responsible for balance and spatial orientation. This meticulous work, elegantly demonstrating the specific function of these structures, had a profound and lasting impact. Flourens's findings directly influenced subsequent research by pioneers like Ernst Mach, who further investigated the physics of vestibular perception, Robert Barney, who elucidated the role of the semicircular canals in nystagmus and developed diagnostic tests for vestibular disorders. The enduring legacy of Flourens's work on the semicircular canals is evident in the continued relevance of his findings to modern ontology and neurophysiology. His careful experiments established the foundational understanding of the vestibular system, which remains essential for diagnosing and treating a wide range of balance disorders. His contributions continue to inform our understanding of the intricate interplay between the inner ear, the brainstem, and the cerebellum in maintaining balance and equilibrium. This work stands as a testament to Flourens's experimental skill and his insightful capacity to interpret complex physiological phenomena. The Flourensian Paradox pioneers resistance to his own discoveries. Flourens's contributions to experimental physiology were undeniably transformative. His meticulous investigations into the functions of the cerebellum and the semicircular canals established foundational principles in neurophysiology. Yet, a curious tension exists within his legacy, a paradox that demands closer examination. While a pioneer in demonstrating functional localization within the nervous system, particularly in the cerebellum's role in coordination, Flourens became a staunch opponent of cerebral localization. This opposition, deeply rooted in his philosophical commitments and institutional influence, casts a unique light on the complex interplay between scientific discovery and personal belief. His resistance to the idea of specific cerebral functions stemmed from a deeply held anti-materialist Cartesian philosophy. He believed the brain functioned as a unified organ, a holistic entity incapable of being dissected into discrete, independent modules. This viewpoint, while seemingly at odds with his own experimental demonstrations of cerebellar localization, profoundly shaped his interpretation of his research in the cerebrum. His experiments, while rigorous, were interpreted through this lens, leading him to downplay or even dismiss evidence that contradicted his holistic view. This is clearly seen in his interpretation of lesion studies, where he observed functional deficits following cerebral damage, but attributed these to general disruption of the organ's overall function rather than to the loss of specific localized capacities. He emphasized the brain's integrated nature, arguing that any localized damage resulted in a generalized impairment. This philosophical predisposition was further amplified by his fervent opposition to phrenology, the then popular pseudoscience that attempted to map personality traits to specific brain regions. Flourens viewed phrenology as a crude and unscientific attempt to reduce the complexity of human mental function to simplistic anatomical correlations. His vehement rejection of phrenology, however, arguably blinded him to the possibility of a more nuanced form of cerebral localization, one that acknowledged the brain's integrated nature while still recognizing the specialized functions of distinct regions. His crusade against phrenology, while ultimately beneficial to the advancement of legitimate neuroscience, inadvertently served to reinforce his own holistic doctrine, potentially hindering the acceptance of more accurate models of cerebral function. The influence of Flourens extended far beyond his experimental work. For nearly 3 decades, he served as the secrétaire perpétuel of the Académie des Sciences, a position of immense power and influence within the French scientific community. In this role, he acted as a gatekeeper, shaping the direction of research and influencing the acceptance or rejection of new ideas. This institutional power allowed him to effectively suppress or delay the dissemination of research that challenged his holistic view of cerebral function. While his intentions may have been rooted in a desire to maintain scientific rigor and prevent the spread of pseudoscience like phrenology, his actions arguably stifled the progress of cerebral localization research for a considerable period. This highlights a crucial aspect of scientific history. The influence of powerful figures in established institutions can significantly shape the trajectory of scientific progress, sometimes hindering the acceptance of groundbreaking discoveries. The Flourensian Paradox, therefore, is not a simple tale of right versus wrong. It is a complex narrative highlighting the inherent tension between empirical observation and theoretical frameworks. Flourens's experimental methods were revolutionary, his contributions to our understanding of cerebellar function undeniable. Yet, his philosophical biases and institutional position led him to resist the very implications of his own discoveries. His legacy serves as a potent reminder of the double-edged sword of scientific authority. The power to overturn established dogma, as Flourens did with phrenology, is balanced by the potential for personal beliefs and institutional influence to create new, equally restrictive dogmas. His story is a timeless illustration of the intricate interplay between empirical evidence, theoretical frameworks, and the institutional structures of science, a cautionary tale that continues to resonate in the modern scientific landscape. It underscores the enduring importance of critical self-reflection, intellectual humility, and the constant reevaluation of established paradigms in the pursuit of scientific truth. The legacy of Pierre Flourens is not one of simple triumph or failure, but a multifaceted reflection on the very nature of scientific progress itself, its triumphs, and its inherent limitations. His life's work remains a compelling case study in the human element of scientific discovery, reminding us that even the most rigorous scientific endeavors are inherently intertwined with the complex tapestry of human thought, belief, and institutional power.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.